Welcome back to the Flagship Studio here at the 2026 JP Morgan Health Conference. I'm joined today in the studio by Steve Harr, CEO of Sana Biotech. Steve, you are focused going forward on two main programs, your diabetes program and your in vivo CAR T program. What gave you confidence to narrow to those two programs? What was the thinking at Sana?
Well, first, thanks for having me back. It's always a treat
you.
to be in the studio, the Flagship Studio, and have this conversation every year. And when we started the company, we tried to go after we thought were two of the biggest areas of unmet need in trying to turn the dream of genetic medicine into a reality. And every time I went to a presentation on cell therapy, it started with a first couple slides saying, boy, if we could just overcome allogeneic immune rejection, we could do so much more. And every time you go to a talk around gene editing tools and say, boy, if we could just deliver these into cells, we could do so much more. And so we thought we would go after them. And as we went after them, there became clear areas where we could deploy the technologies that began to work. And so we love type 1 diabetes because the unmet need is gigantic. It's been 100 plus years.
People, yeah.
Right?
How many people suffer worldwide from type 1 diabetes?
About 10 million people.
10 million people.
I was playing around, like many probably in the listening to this, with one of my favorite chat boxes, trying to put that into scope. And in the United States, if we can cure or functionally cure type 1 diabetes, it would be more or less equivalent on a numbers basis to curing both multiple sclerosis and HIV.
In the U.S. alone?
In the U.S. alone.
Wow.
And so it's sometimes hard to grapple with the size of the problem.
Yeah.
And when you put that into lives lost, if you're in your 20s, a woman would probably rather be diagnosed with either breast cancer or HIV than type 1 diabetes, let alone the day-to-day management challenges. Those are some of the things that kind of came back at me from the chat box.
You're such a good explainer. Do you want to just take a minute to explain allogeneic for people?
Yeah, so allogeneic means someone else's cells. And so our bodies have evolved to really recognize that we shouldn't have foreign stuff in them. We've been attacked by animals and viruses and bacteria and parasites for years. And so if you try to put someone else's cells into your body will see them as foreign, like a virus, and reject them. And so it's been the challenge of transplant medicine now for over 80 years. And so the key in being able to make cellular therapy a reality at scale has been trying to figure out how can we trick the immune system so that it doesn't see these cells as foreign and instead just lets them go about their business.
We come in peace.
Okay, we come in peace. We're here to do good.
So you talked about the narrowing being driven by both scientific promise from your platform and huge impact on people. I want to drill down on the type 1 diabetes. I think our audience will keep in mind, you'd rather be as a woman diagnosed with breast cancer or HIV in terms of cures available. But Newbar's annual letter this year talks about human-made miracles, and where you're headed on type 1 diabetes seems like it's a strong contender for a miracle. Do you want to talk a little bit about what you're seeing in terms of the data there?
Sure, and it is many humans that made this miracle. It's this combination of profound immunology, the ability to gene modify cells with things like CRISPR, and then stem cell scientists. So it takes a village really to make something like this.
Village-made miracle.
Yeah. So type 1 diabetes, actually its etiology is quite simple. It's your immune system has attacked pancreatic beta cells in the body. And the pancreatic beta cell is the only cell that makes insulin. And insulin is what allows us to utilize sugars or glucose as energy. And so about 100 years ago, actually almost 102 years ago to the date, was the first patient received insulin from another source. And it's been, up until then, people died within a few months. And now people can live, and insulin's gotten better and better, and they can live, within about, let's call it a decade of what a normal person, but still a very difficult life, and still there's a meaningful impact on survival. So about 25 years ago, someone realized if you could take pancreatic beta cells from a cadaver and transplant them into a human, they would live. And that these patients could come off of insulin. They lived a relatively normal life, except they had to be on lifelong immunosuppression.
Okay.
And that's not a very good scalable supply source. So over the last, let's call it 5 years, some people have figured out that you can take stem cells, make them into pancreatic beta cells, transplant them, and again, patients do quite well.
Okay.
And that's a more scalable source, but they still have this problem of immunosuppression. And that leads to cancer and infections and a whole host of toxicities. And so what we've shown in this patient, and at this conference we're going to show our 12-month data. We transplanted the first patient. The goal of this study was to see could we gene modify cells and hide them from immune recognition and see them still work.
Okay.
And we're now 1 year out, and for the first time in over 4 decades, this patient continues to make his own insulin. So the cells they evaded the immune system, they survived, and they continue to function. And so now you put the whole picture together, right?
And you're going to share that data on stage here at
We're
JP
going to share
Morgan?
that, yeah.
And that's because Sana has modified these cells to have essentially an invisibility cloak.
Exactly. They're cloaked.
Yeah.
A cloak is a double-edged,
Yeah.
like all cloaks has a little sinister to them, and we have to be very careful with this because the immune surveillance is important.
Yeah.
But yes, that's what we've done.
Got it. Talk to us now about the in vivo CAR T program. Talk about cancer.
Yeah. Before I go there,
Yep.
I'm going to make one other thing.
Sure.
So we've been putting this all together into a gene-modified stem cell-derived islet that we can manufacture at scale. And last year we made a ton of progress, and this year we're going to put that into human testing, start our phase 1 study.
Okay.
And we think there's a very rapid path to proving that this works. And once it works, hopefully, as we figure out how to scale manufacture even more, there's a relatively straightforward path to bringing this to patients as a commercial product.
Do you expect to have shareable phase 1 data in 2026?
We're not sure.
Okay.
It's called 12 to 18 months.
12 to 18 months,
Yeah,
okay.
it's in that range.
Gotcha. Okay.
With the in vivo CAR T cell, we do expect to have data this year.
Okay.
So the idea of CAR T cells have had a tremendous impact on the treatment of blood cancers. They look like they may have a tremendous impact on a host of autoimmune diseases. And they are a one-time therapy that can lead to kind of a curative-like phenotype for patients. The challenges have been it's super difficult to scale them. Patients have to take high-dose chemotherapy, reasonable dose chemotherapy before they take them. And with the in vivo CAR T platform, we're optimistic we can overcome both of those.
Wow.
There's been some progress in the field that suggests that that's likely to happen. And we think we have the potential to have a best-in-class therapy. We made 2 really fundamental bets when we did this. One is relatively obvious, I think, but not something others have done. That is that cell specificity matters when you do this. You want to deliver this genetic payload only to the cells that you're targeting, not to a whole bunch of other ones.
Address, yeah.
And that has a whole host of benefits. The other is that you have to get this DNA into the genome of the target cell because it has to divide logarithmically, and you need the progeny to have the same signal, right? If those 2 things turn out to be right, we've done a lot of work. It may be simpler ways to solve it, but those 2, if they are right, then we think we have a high probability of having a best-in-class therapy. So we hope to start studies this year. We hope to have data before the end of the year from the first drug, and the team's made a lot of progress.
In vivo CAR T seems to be an area that's heating up in a sector that is finally reheating up. Lots of activity in biotech in the merger and acquisition space, the partnership space. What are you looking for from the JP Morgan conference this year, and what are you looking for from 2026 for Sana in terms of markets, investors, partners?
Most important are investors. We are trying to build a sustainable company that has the potential to deliver these medicines to patients. And in particular, the complexities of type 1 diabetes are novel, and so it isn't clear to us what large companies may help us move this forward faster. Right? That's ultimately the goal of partnerships, is to move it forward faster and deliver it to more patients. Right? And so we need capital to make that happen, and our goal is to continue to attract the capital to allow us to push the type 1 diabetes forward. In the in vivo CAR T space, it's a very competitive area, and essentially every big company you can think of has some way of targeting B cells and plasma cells, and they're utilizing that for cancer or for autoimmune diseases. And so we have to be fast, right? And we have to do things at a rapid scale. And so thinking there that about a partnership is much more reasonable to us, because they can really help us get this medicine to patients hopefully faster in a broader way as well.
Steve, thank you so much for joining us in the studio. Sana Biotech bringing us village-made miracles faster for cures. Thanks so much.
Always a pleasure.